As precise and predictable as the rules of genetic expression seem, permanent changes do occur in the genetic code. Indeed, genetic change is the driving force of evolution by adding variation to population of organisms. Changes may become evident in altered gene expression, such as in the appearance or disappearance of anatomical or physiological trait. For example, a pigmented bacterium can lose its ability to form pigment, or a strain of the malarial parasite can develop resistance to a drug. So a phenotypic change that is due to an alteration in the genotype is called mutation. So this is what you're going to learn for the next few slides. In short, mutation refers to a heritable change in the genetic material. So mutations are changes in the DNA that results in different characteristics. So mutations can occur by either addition of bases or deletion of the bases or replacing of the nucleotide bases. So this is the consequences of the mutation. So what you have is a difference in the nucleotide base sequence. And then if you have a different nucleotide base sequence, then you will have a different sequence of amino acid. Then that difference in amino acid will create a different polypeptide, and so you will have a different type of protein or different protein with different characteristics. So therefore, finally it gives change, or it results in changes in the organism characteristic. A microorganism that exhibits a natural, non-mutated characteristics is known as the wild-type strain or the wild strain. So if microorganisms bears a mutation, then it is called a mutant strain. So these mutant strains can show variants in morphology, nutritional characteristics, genetic control mechanism. So they can also be resistant to chemicals, difference in temperature preference, and nearly any type of enzymatic functions. So in order to understand how a change in DNA influences the cell, remember that the DNA code appears in particular order of triplets, so the 3 bases that is transcribed into the mRNA codon, each of which specify an amino acid. A permanent alteration in the DNA that is copied faithfully from mRNA and translated can change the structure of a protein. A change in the protein can likewise change the morphology and physiology of a cell. So most mutations have a harmful effect on the cells, leading to cell dysfunction or death. So these are called the lethal mutations. Neutral mutations produce neither adverse nor helpful changes. A small number of mutations are beneficial in that they provide the cell with a useful change in the structure or physiology. Cells have a very good DNA maintenance system so that the mutation rate are extremely low. One mutation happen out of 10 to the power of 9 nucleotides per generation. So that's why for mammals and for human the mutation rate is very low, but not so much on viruses. So virus is highly mutated organism. As I mentioned before, so the mutation can actually create or can be useful for modification of proteins. For example, mutation or protein engineering uh, is done in order to increase the stability of the protein. So for example, increase in the thermal stability so that it can withstand high temperature, useful for the industry. And then also it can increase the stability at low or high pH values, or you can make it resistant to oxidative inactivation. And also you can create or mutate the protein in order for it to have a very broad spectrum of activity and alter the substrate specificity. A mutation is described as spontaneous or induced, depending upon its origin. So a spontaneous mutation is a random change in the DNA arising from errors in replication that occur without a known cause. So the frequency of spontaneous mutation has been measured for a number of organisms. Mutation rates vary from one mutation in 10 to the power of 5 replication. So this is considered as high rate to one mutation in 10 to the power of 10 replication. So this is in the case of a low rate of spontaneous mutation. So the rapid rate of bacterial reproduction allows this mutation to be observed more readily in bacteria than the eukaryotes. The second type is the induced mutations. So these mutations result from the exposure to known mutagens. So these mutagens are primarily physical or chemical agents that damage DNA and interfere with its functioning. The carefully controlled of mutagens have proved a useful way to induce mutation or to induce mutant strains of microorganisms for study. So these are the common causes of mutations. For spontaneous mutation, you have the aberrant recombination, aberrant segregation, error in DNA replication, toxic metabolic products, transposable elements. Also, we have depurination, deamination, and tautomeric shift. For induced mutation is caused by either chemical agents or physical agent. These are the selected mutagenic agents and their effects. Chemical agents such as nitrous acid and bisulfite. So this compound will remove an amino group from some bases, and then we have ethidium bromide. So ethidium bromide will insert between the paired base bases. Acridine dyes will cause frame shift due to insertion between the base pairs. Nitrogen base analog will compete with the natural bases for sites on the replicating DNA. For the chemical agent, for the physical agent, so radiation normally cause mutation. The ionizing we have ionizing radiation such as gamma rays or X-ray. So these will form free radicals that cause single or double breaks in the DNA. UV light will also cause mutation. It causes cross-links between adjacent pyrimidine. Chemical mutagens come into three main types, how they alter the DNA structure. So either they are the base modifiers, they are intercalating agents, or the base analogs. Base modifiers covalently modify the structure of a nucleotide. For example, the nitrous acid will replace amino groups with the keto groups. So this can change a cytosine to uracil and adenine to hypoxanthine. So this is how it looks like, the changes that happen when nitrous acid is present as the mutation. So you have cytosine here. The amino group in the cytosine will be replaced with the keto group. Okay, so cytosine is changed to uracil after replication. And for adenine, so the amino group here is changed to a keto group here. So therefore, there's no more adenine. Then the basis is now paired with hypoxanthine. So this is called the mispairing of the modified bases. The other type is the mutation as the intercalating agents. So these intercalating agents contain flat planar structure that intercalate or come in between. Uh, so intercalate themselves into the double helix. So this will distort the helical structure. So when DNA containing this mutation is replicated, the daughter strand will then may contain single nucleotide additions and or deletion. So examples of the intercalating agents are the acridine dyes, proflavin, and ethidium bromide. So another event or type of effects of the mutations is the base analogs. So base analogs become incorporated into the daughter cells during DNA replication. So for example, 5-bromouracil being incorporated into the DNA. So 5-bromouracil is a thymine analog, so it is the same as thymine. So normally thymine will pair with adenine. So instead of the normal thymine will pair with adenine, this base analog 5-bromouracil will pair with adenine. So therefore, the bases are different in the DNA strands. So also you have a tautomeric shift occur at a highly relatively high rate, whereby this 5-bromouracil can also pair with guanine. Okay. Physical mutagens come into two main types. Either they are the ionizing radiation or the non-ionizing radiation. The ionizing radiation includes X-rays and the gamma rays. So these rays have short wavelength and high energy. So with that high energy, so this ray can penetrate deeply into the biological molecules. This will create chemically reactive molecules termed as the free radicals. So these free radicals will can cause either base deletions or single nicks in the DNA strand. So also cause cross-linking and chromosomal break. The non-ionizing radiations include the UV light. So it has less energy. It cannot penetrate deeply into the biological molecule. So it causes the formation of thymine dimers. Okay, thymine dimers, and these thymine dimers may cause mutation when that DNA strand is replicated. So here, when there is UV light, then cause this thymine will link to each other, then resulted in a thymine dimer. So you have TT. Now let's have a look at the categories of mutation. Mutations range from large mutations, in which large genetic sequences are either gained or lost, to small ones that affect only a single base on a gene. So the small mutation involve addition, either addition, deletion, or substitution of single bases. So the small mutation is called as point mutation. So a point mutation is a change in a single base pair. So it involves a base substitution. So a point mutation can be either a transition, so where a change of a pyrimidine, either cytosine or thymine, to another pyrimidine. So for example, C to T or T to C. So or purine to another purine, A to G or G to A. Another type is the transversion. So transversion is a change of purine to pyrimidine or vice versa. So transition, the type of mutation, the transition one is the more common than the transversion. Other than the base substitution, mutation may also involve in the addition or deletion of short sequence of DNA. For example, here we have 2, 4, 6, 8, 9, like 10 or 11 nucleotides here. So these 4 nucleotides being deleted. So for deletion of the 4 bases, base pair will result in a shorter DNA sequence. Or you can also have the addition of 4 base pair from this sequence here. So you have so that resulted in a longer um, nucleotide sequence. Mutation in the coding sequence of a structural gene can have various effects on the protein or the polypeptide. So any change in the code that leads to placement of different amino acid is called a missense mutation. So missense mutations are those base substitution in which amino acid change occur. So a missense mutations can do either one of these. So either they can create a faulty non-functional protein or less functional protein. So or it can produce a protein that functions in a different manner, or it can cause no significant alteration in protein function. So if the substituted amino acids does not affect any protein function, so as measured by phenotype, so the mutation is said to be neutral. So the nonsense mutation, on the other hand, changes a normal codon into a stop codon that does not code for an amino acid and stops the production of the protein wherever it occurs. So a nonsense mutations almost always produce or results in a non-functional protein. A silent mutation alters a base but does not change the amino acid and thus has no effect on the protein. So these mutations are those base substitutions that do not alter the amino acid sequence of the protein. So this is due to the degeneracy of the genetic code. So you know that the code for amino acids, for one amino acids, can have a few codons that encode for that amino acid. For example, for threonine, so it has for example, ACU, ACC, or ACG and ACA that code for the threonine. So a mutation that change only at the last base will not alter the amino acid. So for threonine, the first 2 letters are the same, and either one of the letters at the back, the third nucleotide, if it's changed, it will still code for threonine. So that is called the silent mutation. So mutation also occur when one or more bases are inserted into or deleted from a newly synthesized DNA strain. So this is called as frameshift mutation. So frameshift mutation involve the addition or deletion of nucleotides in multiples of 1 or 2. So this shift the reading frame so that completely a different amino acid sequence occur downstream of the mutation. So let's have a look at in detail how does all these type of mutations are? What are their characteristics? So these are the consequences of the point mutation within the coding sequence. We have the different type of the of change. For example, we have the if it's non-change, so you don't have any mutation. So this is how the DNA sequence look like, and also this is how this genetic code will produce the amino acid. So this is the sequence of the polypeptide. When there is a silent mutation, so it involves a base substitution. So what happened is that if you notice here, so this here C, so proline is encoded by the codon of CCG, and also it is encoded by the codon CCC. So substitution of the third base does not change the amino acid. So it still code for proline. So that means it is a silent mutation. There is no changes in the protein sequence. The missense mutation is a type where involves a base substitution. Missense is normally will affect the amino acid sequence and further down the physiology and so on. So for example, methionine, you have ATG, and then you have a change in the first base of the codon for threonine here. So we have initially it is ACC, right? Now A is changed to C, so now it become proline. So all in all, the sequence of the polypeptide is now changed. Okay, so that is the characteristic of the missense, or the consequences of the missense mutation. The nonsense mutation will create a base mutation that leads to a stop codon. So here we have AAA here that supposedly encode for lysine, but instead the A here is changed, is substituted with T. Then you have here a stop codon, so TAA. So that means the protein production, protein synthesis will stop, so you will have truncated protein. So this will lead to definitely a non-functional protein. A frame shift mutation, it actually brings either a process of addition or deletion of the nucleotide. So the consequences is that, for example, here you have an addition of G here in between the C and so you have an addition of G here in between C and C here. So that GCC will so the amino acid will change. Instead of proline, and then you will have this three nucleotide will code for alanine. When this frame shift mutation happen, when one nucleotide is added, so you have a frame shift. The codon will shift, right? The reading of the codon will shift. So it affects the rest of the amino acid sequence. So now you will have a completely different amino acids right after the addition or deletion. Earlier, we indicated that DNA has a proofreading mechanism to repair mistakes in DNA replication that might otherwise become permanent. So because mutations are potentially life-threatening, the cell has additional systems for finding and repairing DNA that has been damaged by various mutagenic agents and processes. So most ordinary DNA damage is resolved by enzymatic systems specialized for finding and fixing such defects. So these are examples of the enzymes that help in the repair of mutations. So they are the DNA polymerase is the one that proofread nucleotides during the DNA replication. So also we have a mismatch repair process whereby the enzyme will locate and repairs mismatched nitrogen bases that were not repaired by DNA polymerase. And we have also light repair for any UV light damage, that any mutation that caused by the UV light damage. And also we have excision repair, so where this system locates and repairs incorrect sequence by removing a segment of the DNA and adding the correct nucleotide. So it is amazing how cells work, so to maintain the sequence of the DNA.